So welcome to this session. Good morning, good afternoon, and good evening, wherever you are. So in this session, we have two lectures. First one will be delivering by Professor Roop Mallik. Professor Mallik started his research career in theoretical physics and obtained his PhD from
Theoretical
PhD.
physics? I thought
No,
he's an
no,
experimental.
objection, Your Honor. Experimental physics. I've always been an experimentalist.
Okay, so experimental physics and from TIFR. And after staying a brief stint at TIFR as a postdoc, and then he moved to UC Irvine, and from where he started his work on motor protein. So currently he is a professor at Bioscience and Bioengineering Department at IIT Mumbai, and the title of his talk, today's talk, is on and off controls within dynein and dynactin on nature curve. So yeah,
Okay.
Professor Mallik, screen is
So
yours.
let me just share my screen. 1 minute. Can you see the screen?
Yeah, I can see you.
Okay, great. Okay, thank you. And thank you, Shudipto, and good morning to everybody. And I will take the next hour to tell you about some work that is in progress. But I realize that for the more diverse audience that we have on this kind of a talk, I should give you a little bit more background about things that we have done and about. I particularly wanted to give you some more background on how these kind of experiments are done. And we have heard, in the opening talks by Steve Gross and Vaishnavi. So I guess you people have some background, but I will remind you about a few things and how things are done by others and how we do these kind of experiments. So the title of the talk is On-Off Controls within the Dynein-Dynactin Machine, and I will explain these terms. But before I go into those specifics, I will talk about some earlier work, published work, which we have done. And I would like to acknowledge Paulomi and Pankaj, students in my lab at TIFR, who have contributed very well to this story, which you see the title for here. And I would also like to acknowledge Ankit and Madhusudan at IISc Pune, who have helped with certain aspects of this work. Now let me go into the laser pointer. Yeah, right. So motor proteins and transport of cargoes inside cells. As I just told you, we have heard about this from Steve and Vaishnavi earlier. And oops, what was that? Hold on, something happened. Sorry. Yeah. Okay. So if so, what we do know from work. Sorry. Just hold on. Give me a minute, yeah. Right. So what we know from work by many other people in the past is that things like this happen inside cells, where here you are seeing a pigment granules inside cells which kind of live right under your skin and which help you, change color if you are a chameleon, or be darker in color than other let's say Caucasians, like we are darker in color here in India. Now each of these little balls that moved around, in this case, in this specific case, what you saw here actually moves on structures like these, which are fluorescently labeled inside a cell, and you can see these filament-like structures. These are the microtubules. And they are shown again in this animation here, which I'm sure many of you are familiar with. And here is a single microtubule, which is inside the cell, picturized here inside the cell. And there are some actin filaments shown here. And on such a microtubule, you have, the picture is that you have these kind of vesicles, in this case, these little pigment granules, being carried by motors of different kinds. In this case, shown here is kinesin. Now, this movie is a bit misleading in the sense that this is not very often from what we have now started to understand, this is not how things work. And the situation is far more complex and actually far more exciting, at least to me, than what you saw in this animation. So, so the reason we have this kind of a picture, to my belief, is because a lot of the studies that we have seen over the last 2 or 3 decades have been with highly purified systems, where you can purify these motors or these some form of this cargo, artificial or not so artificial, from the cells, and assay how they work and measure their movement with pretty high precision in a reconstituted system where you do this on a cover slip. But that's not something. I mean, that's the kind of work I had done in my earlier tenure, but that's not what I wanted to do when I came back here to India. And we wanted to do something which would tell us more about what happens really inside cells and how these machines work. And so we took rather than go take a bottom-up approach where we try to reconstitute everything on a cover slip, we took a little bit of a top-down approach where we tried to extract cargoes, real cargoes from inside cells, and also study them directly inside cells, and put these cargoes on a cover slip and see them move. So the way we did it, this is largely published work, but I will just quickly go through it, is that you take polystyrene beads, plastic beads here shown in blue, and let's say they are 1 micron in diameter. You can take slightly smaller or larger ones, and you grow cells. Doesn't matter what kind of cells it is right now exactly. You can grow cells on a cover slip shown like this, and you add these beads to the cells. And these many of these cells, the kind of cells we use, like to eat foreign objects. That is because they're part of your immune system, and they're constantly clearing different kinds of foreign objects, let's say benign, particles which you inhale, or in fact, many kinds of pathogens. And this is what makes it really exciting. Many kinds of pathogens are actually ingested by these cells. And when that happens, the question is what happens to these pathogens inside the cells? Because you understand that normally most pathogens will get cleared by your immune system, but many don't. And so the trafficking of these pathogens, where they end up at certain points of time inside the cells of your immune system, is extremely important to understand. And that's something we have been trying to do, although I won't be talking about that much. So you feed these beads to the cells, then leave them for a while so that these cells get eaten up by, the beads get eaten up by the cells. And this is what you see after some time. This is one single cell. I don't have a scale bar here, but each of these beads is 1 micron in diameter. So you know how large this is. And you let them be for a while, and then you take these cells and crush them, and you can simply take this through a centrifuge, and all these beads, because they are extremely buoyant, the density is lower than the watery medium in which they exist, and you can just float them to the top and you can extract them. Okay, so then what you get if you extract these objects from these cells is not this. This is the classical view of a single motor, which has come by because of these assays where you coat beads with motors. So you don't get this. What you get rather, although you have a bead inside, which gives you many advantages in doing these kinds of biophysical assays, which I will come to, instead of what you see here, what you get rather is an object like this. Okay, what is shown here on the left hand side, and this is actually what this means is that this cargo, the bead, now we will call it a phagosome. So just remember this word phagosome, which has been extracted from the cells. Basically, inside this phagosome is a bead, and outside is all these proteins which are on the surface of this bead. And these proteins may include the motor proteins, the dyneins and the kinesins, and that's what you are seeing in this picture here. So ignore this kind of ellipsoidal shape. Imagine it to be a spherical if you have a bead inside, but you have all these proteins stuck on the surface, which have been assembled there by the cell, not by you. And you have these different motors shown red, shown, one class of motors which goes towards the center of the cell and the other which goes towards the periphery, the dyneins and the kinesins. Very importantly, something that I will again not talk about today is another component which you have in this assay, which is a lipid membrane, which you kind of see here in this greenish color here, on which all of this, motors and other proteins are assembled. That we are now find is extremely important to understand the biology as well as the physics of this system. So once you have such a system where you have purified such a cargo from cells and you have these motors, so you have to now think of not in terms of a single motor, as I have depicted in this picture. You have to now think of in terms of probably more larger numbers of motors trying to drive a cargo, in this case this phagosome, towards the plus end or the minus end of the microtubule. And very exciting questions arise about how these kind of motors, as you have seen in this picture, they have these arrows are pointing in different directions. They are very often antagonistic motors that they exert forces in opposite directions. How these, how does this cell use this antagonistic activity of these motors to localize things inside the cell? This is the fundamental question which many people are trying to understand. How does how do things get localized at the right place because of this opposing activity of motors, and how does the cell control this opposing activity to take something outside or take something inside? Okay. So again, just to, a little bit of a recap, the way these experiments have been done is that you have this cargo here. In this case, imagine a single motor on this, and then this motor, here is a cover slip shown here, and on that there's a microtubule stuck. And from the bottom, you shoot a laser light through the objective lens of a microscope so that it focuses in a tight spot here. And typically, these plastic beads have a higher refractive index than the medium around it, which is like water. And so you can exert a force, and this is basically an optical trap. And the position of this bead in this, in this trap with respect to the center of the trap can be determined with very high accuracy using a detector arrangement, which I'm not going to go into right now. So essentially what happens that if I look at this object here, a bead with a motor on it, this is what happens. Here it comes along and suddenly you switch on the trap, and this bead, the motor tries to pull the bead out of the trap. It goes for a certain distance, falls back, again tries that, falls back again. And this, sorry, I forgot to mention here, this is taken from George Shubeta's lab, this cartoon. But now the excitement is that you have to imagine a situation where instead of a single motor, instead of this object, you have a more complicated object which is extracted from the cells, this phagosome, and you have teams of antagonistic motors trying to pull this cargo back and forth in the optical trap. So how does this dynamics work out? And if we understand this dynamics, can we understand something more about how something gets localized to a particular place inside cells? Okay. So this kind of assay, of course, we do many other things, which maybe I will not have the time to go into. Not optical trapping is not the only thing that you need to do to understand this. This kind of an assay gives you this kind of data, and this is important for us to understand. So let us look at it closely. On the x-axis is time, and on the y-axis is this distance from the center of the trap to the instantaneous position of this cargo. It could be a phagosome or a bead. So in this case, let's say you have a single kinesin motor pulling you away from the center of the trap. So this distance is on this y-axis here. And as you go farther, you go for some distance and then you detach. And that's what happened here. And then you can try again and again and again. So by measuring this distance, if you know the stiffness of this trap, So this optical trap actually works like a spring in the sense that if I move something out of the trap pulls it back in, and you can show by ways I'm not going to go into that this works like a linear spring within certain region. Okay, so you can now, by multiplying this distance on the y-axis by the stiffness of this trap, which you can calculate by separate experiments, you can find the force that is being exerted instantaneously. And from that force, most interestingly, you can also calculate the number of motors which are active at a certain point of time. Now, to me, this is really exciting, particularly if you have such a cargo in which you have these antagonistic motors, unknown numbers of them usually, pulling and pushing against each other. So you can, what you can do is that you can measure the kinetics of this process in real time with very high resolution, and this is not usually possible, in most systems. So if you get inside a cell now, right? So when you enter a cell, let's say that there's that bead entering the cell, what happens is that initially these beads, do this kind of fluctuating motion where they are moving along these microtubules, and this we call an early phagosome. And while they are moving, they are undergoing cellular, cell-driven changes and chemical changes in the membrane and in the proteins, and they end up after some time, 10 to 15 minutes, becoming a late phagosome. So initially you were early and you did this kind of a dance, back and forth dance. And then while you were doing this dance, you were changing. And then you become such an object which is now yellow, showing that it is different, a late phagosome, because it has spent some time. And when the late phagosome moves, please move, yeah, you see that the motion is very different in the sense that it more or less unidirectionally it goes towards the center of the cell, and this motion is dominated by dynein. So what I will do today, I will try to give you examples of one kind of motion, which is this back and forth motion, the work that we did on that and how we analyze that data. And then I will come to this late phagosome, how you can use the advantages of this system to understand intracellular transport. So the experiment, again, just to repeat, is here you have such a phagosome. It is pulled by these kind of teams of antagonistic motors, and you are measuring its position on the microtubule with respect to center of the trap. So if you take an early phagosome, remember the green object in the previous slide, it moves in a back and forth manner. So here is an early phagosome, EP. What is this data? It is a single phagosome held in this optical trap, and sometimes going towards the right, sometimes going towards the left, shown here as up or down. We know, because we know the polarity of this microtubule, one side is plus end, which is towards the periphery of the cell, the other is minus, which kind of, if you go below zero, then then this activity is driven by dynein. If you go above zero, this activity is driven by kinesin. Okay, so here is a system where you can in real time really measure how these motors are functioning and record that very nicely. Of course, if you do the same experiment for late phagosomes, the yellow one, which moves towards the center of the cell, you see a very different kind of data where most of the action is downwards, means that it is driven by dynein. So this is, you can look at the schematic at the right here. And if you go down, your dynein. If you go up, your kinesin. So you can now you can see that the motion is dominated by dynein here with intermittent kinesin driven motion. And that's understandable because you want to go towards the center of the cell where you want to fuse with lysosomes. So what we did is that with help from Ranjit, and I will come to that part in a bit later, is that we could measure this kind of activity and analyze it. And so if you go up, your kinesin, so your K. If you go down, your dynein, then your D. So you see a reversal here, right? You had kinesin and then you went down, so you had dynein. And then you had dynein and then you went up, you had kinesin. So you call this reversal a KD reversal. Kinesin to dynein, DK, dynein to kinesin, and so on and so forth. You can, of course, have kinesin because you didn't go the other way, or you can have dynein because you did two successive stalls towards the minus end of the microtubule. Now, you can count simply you can take this data for a long time and you can count the number of KD and DK occurrences, and you see that all of them occur with more or less equal probability. Okay, so the number of DD reversals this is equal to the number of DK, KK, KD. So what this means is that if you are in a given state, let us say here, you have equal probability of going to the same state, being driven by kinesin after this, or going in the other state, which is driven by dynein. And this obviously sounds familiar, I'm sure, to all of you. And this is similar to what you would expect if you were tossing a fair coin. And so if you imagine dynein as tail and kinesin as head, so head, 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 tail, 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 head. So these are the four possibilities if you look at the pair events, and you would get 25% probability of having all of these. So this data represents or rather is similar to what would happen if you were to toss a fair coin. Now the interesting part is that this, we feel, has a fairly deep implication, because these early phagosomes really want to move around and go nowhere. Okay, they want to fuse around with other organelles. So you adjust the activity, and when I say adjust, I don't want to get into the details of that, but you kind of accommodate the activity of the kinesins and the dyneins so that they have equal chance of getting activated. And so that is then a fair coin, right? So you can have a head or a tail at every toss with equal probability, and so you get this kind of a behavior. Now, is there a way in which you can make this coin unfair? So you either bias the dyneins or the kinesins, and if you can do that, then you can take this cargo outwards or inwards, depending on which one you're biased. And we actually showed that this can be done. It is actually done on the late phagosomes, where these dyneins and the kinesins organize in a different geometrical manner on the surface of the phagosome, and that biases this coin towards an unfair coin favoring dynein, and then you go towards the minus side.
There is a hand raised, so can you?
Yes, I can take a question if there is one.
Yes, I have a question.
Please.
When you are plotting these position versus time data, we can also see some zones which are like plateau, where the position is not changing with time. So those are the regions which
I'm coming to that. I'm coming to that. So you can see two kinds of plateaus. One plateau is where you are at the center of the trap. Okay. Hello, can you hear me?
Yes.
Yeah, one plateau, if you can see my cursor, is at the center of the trap, is at the zero position, where basically nothing is happening because no force is being generated. And another plateau is away from the center, where the motors have exerted force, but they can't win against the trap anymore. So you are horizontal. Okay. So these are inactive regions and these are active regions where you have stalled. Does that answer your question, or did you have a different question?
Yeah, yes, I was asking that when you say stalled, that means an equal and opposite force is being exerted by the trap plus the dynein. Is that correct?
The trap, so dynein is trying to pull you out and the trap is trying to pull you back.
Right.
Yeah. Can I go on?
Yeah.
Yeah, thanks.
Yes, thank you.
Okay, so somehow I can't, So now what we did is that we analyzed this data, and I'm going to go through this quickly because I want to talk about something else. One thing that we focused on was this time, which is shown here in yellow. This is basically these regions where nothing happens. Okay. So what is the distribution of these times, inactive times, before one or the other class of motors gets activated? Okay. So you can measure these in lots of this data. So you can take this one and this one and this one and plot a histogram of these inactive times. So what you have here on the x-axis is this time, a histogram of that time taken across DK events, where you went from here. So for example, this one is the inactive time for a KD event, because you were kinesin and then you went to dynein, and this inactive time, and this is plotted here. Sometimes it is short, sometimes it is long, etc., etc. And from help with Ranjit, what we were Ranjit Padinteri, what we were able to do is that we were able to show that given the known single molecule on and off rates of kinesin and dynein, these would engage within a time of roughly 0.5 seconds to the microtubule if the cargo is held close to the microtubule, which is happening in this case because of the trap. So, this, if the kinesins and the now you have to imagine that here is the cargo sitting on top of the microtubule, and there's a red patch on this. Imagine a red patch. I mean, it's not actually red, but imagine a red patch from where, if you are a motor, you can reach the microtubule. If you are outside the red patch, you will not be able to reach because you're not just long enough. Okay. So then the question is, if these motors attach and detach with known rates, and if we know the area of this red patch, which you can calculate by simple trigonometry, then what is the probability of and if these motors are attaching and detaching independently of each other, do you expect these kinds of plots? Okay. Now to kind of understand this, we gave them enough time to bind, which is this 0.5 second, which is what I told you. And we look at the geometry of this contact and the density of kinesin and dynein motors on the on this object, on this phagosome. And this is, I think I will skip this. You can calculate this area. But basically, what you know is that you know the number of motors which are active because you can measure the forces, right? And you know the number of motors which is active for the kinesins, and it is 1 to 2. And you know for the dyneins, the number of motors that is active is usually 6 or more dyneins are active. So then the question, simple question is that, given a surface density of these motors, which we calculated or actually estimated from experiments, and the typical time which they take to bind and unbind, what is the probability of finding more than 6 dyneins? Why do I say more than 6? Because that's what this force histogram tells us, or more than one or two kinesin within this red patch from where it can contact the microtubule. And you can actually calculate these probabilities because you know the surface density and you know the area within which you want it to localize, so you can get a probability out of that. And so the joint probability of finding 6 or more dyneins in that red patch here, imagine, and this 1 or more kinesin, you can just put in the binomial, do a binomial calculation to find that this probability is 74%. And that is really interesting because that's exactly what you see here. Okay, so you get the probability of this an immediate reversal. Okay, if you were kinesin and then immediately go in the opposite direction, that would happen if you had both motors sitting right there and you don't have to rotate. You can immediately go in the other direction. That is about 75%, which matches really nicely with this number. Okay, so for this part, the take-home message is what we believe. I've not shown you all of this data, so if you are interested, please go and take a look at this paper, cited down here at the bottom, that the activity of opposing motors can be explained by a stochastic mechanical computation, and it appears to follow the tossing of a coin. So you have to, this is kind of a cartoon where you toss a hypothetical coin and you decide whether you're going to go dynein or you're going to go kinesin. And the more exciting part to me is that there are, of course, as I showed you in the earlier picture, along with these dyneins and kinesins, there are so many other lipids and proteins associated with them. They are likely biasing this coin towards kinesin or towards dynein. And by doing so, they can dictate where you will end up inside the cell. So this might be a way of understanding how to localize things and to generate organization at the cellular scale. So I will move on to what I had given as the original title of my talk. And this background that I gave you is useful because now we are familiar with this object which has been extracted from the cells. As I told you, this is this phagosome. And again, I apologize for this ellipsoidal picture. Imagine it as a sphere. And here you have this teams of dyneins and kinesins. And we will now focus on the dynein motors. And dynein is interesting because it has this partner. Okay, so what you see in this cartoon, so you have to imagine dynein walking along a microtubule here, And on top here is a cargo, which you are not seeing in this cartoon, this cargo basically. And dynein walks along, and dynactin, this object here, the gray one, is another protein called dynactin. This is really exciting, interesting, this molecule dynactin, because for almost all known functions of dynein inside cells, you need dynactin. Okay, if you mutate dynactin and make it dysfunctional, then dynein doesn't work inside cells. And the kind of a picture, this animation is quite nice because it seems like it kind of helps dynein in some way by attaching to it, but we don't know exactly how this works. And there are many mutations in this protein dynactin which are known to cause fairly severe neuronal diseases. So there's a lot of clinical interest in understanding, how this whole thing works. Now, to understand this, there are several people who have contributed to this. I apologize because I won't have the time to go into all the details today, but broadly, you can the way people have tried to understand this, you can categorize into two classes, where people have taken a very in vitro approach, where you basically purify dynein either from bacteria or from some other source, and you purify dynactin, okay, or fragments of dynactin or fragments of dynein, etc., etc., and you put them together in, along with other purified components on a bead or an artificial cargo, and then you try to see how this thing moves if I remove this part or if I remove this interaction, etc., etc. And another class of studies where people have looked inside cells, trying to understand how this machine works, where you can mutate, let's say, this part of dynactin, or you mutate something here, or you mutate something here genetically, and try to infer the effect of that. Now, we wanted to be somewhere in between these two extremes, where you have a very artificial system, and it is, one has to be really careful about interpreting those studies. Because all these are purified components and we don't really know whether they are actually attaching to each other in the way we think they are. And the other extreme is the cell culture studies, where yes, you see the real thing, but it's difficult to interpret this data because there are just so many confounding factors. So we thought, why not use this phagosome system, where at least at the cargo level, we are fairly sure that things are like normal inside cells, but once you have extracted it from the cell, all the other complexity is removed. And if all the regulators, all the things which control this to a large extent are resident on the cargo, then we can understand this better. So the picture that has emerged from work, these kind of in vitro work and work inside intracellular work, is something like this. And this is also interesting because most of the work is in the context of neurons, because this, as I told you, mutations cause neuronal disease. So you have these microtubules that I showed you earlier. They can be tyrosinated, which is basically addition of a chemical group, or not tyrosinated, and this tyrosinated versus detyrosinated is different in different parts of the cell. So the nucleus of the cell would be somewhere here, and this is the periphery of the cell. So you can see that when you go towards the periphery, you're tyrosinated, and when you go in, you are less so. So, what has been, what people believe is that this dynein-dynactin complex assembles on a cargo by various means. So this is dynactin here, look at this one, and this is dynein in blue here, which is this one. And this cargo, once it is assembled, has an affinity for this tyrosinated, the green microtubules. And so this cargo with the dynactin comes and quickly sits at this end. And it doesn't sit here because it doesn't like this detyrosinated one. And you want this cargo to sit here at the periphery, because then you can go inside. Okay. So now, also, recently what it has been shown is that each dynactin molecule shown here, so dynactin is this object here, is attached, recruits two dynein motors. So here is a more real version of that cartoon that I was showing you earlier, that imagine each dynein with these two heads, like your two legs. So whatever you have here is shown in this picture here. And these, this can somehow in some manner probably coordinate to walk inside on the microtubules. So what we wanted to do is that we wanted to use this phagosome system, which I have been talking to you about, and we believe, I won't go into those details, that we have really intact dynein-dynactin complexes and indeed kinesins sitting on this cargo on a lipid membrane. And now if we have such a phagosome, and look at this animation, but you have to imagine a phagosome, a cargo above this large one, which is not shown in this. And if I can now add something to the cover slip. Remember, this experiment now we are doing in a cover slip where we have extracted this phagosome or this cargo from the cell, and we are now trying to see how the dynein and the dynactin works. And we know that the motion is driven by dynein or dynactin because we know which way it is moving towards the minus end of the microtubule in this case. So what we thought we will do is that we will add a small peptide. A peptide is a part of a protein, and this peptide is that part of dynein which binds to dynactin. Okay, so this small artificial peptide is the part of dynein which binds to dynactin. One part of dynein which binds to dynactin, and if we do that, then if you have lots of this peptide floating around in the solution, the peptide should go and get stuck to the dynactin, and then it should remove the real dynein from this. Okay, so this is a competitive assay simply where you disable one interaction. And also what we thought we will do is that you can make antibodies against this part. Imagine dynactin's skates. Okay, if dynactin has these skates by which it attaches to the microtubule, I can make an antibody against this region of dynactin. And so it should go, and if I add it to the cover slip, it will go and get stuck to this part. And then this guy, dynactin, cannot bind to the microtubule. So by doing so, you can try to address two questions. One is, what is the role of this binding between dynein and dynactin? And two, why does dynactin need to bind to the microtubule? And this is very interesting because dynactin is the only known associated protein associated with dynein that separately and independently binds to the microtubule. So with this kind of situation in mind, where you don't do any perturbation, or you disable this interaction, or you disable this interaction, we extract these phagosomes from the cells and do this assays, the optical trap assays, and analyze this data. And along with this, we do a lot of biochemistry and some lipid membrane reconstitution, which I may not have the chance to go in today. So the first thing, let us look at what happens if we add this little peptide here. We will call this the dynein intermediate chain, DIC, because that is the part of dynein which binds to dynactin. So if we add that, these two guys should not talk to each other. Okay, and this strategy has been adapted by others earlier, and I've cited this here, but that was not done with this kind of cargo. So the first thing is that if we add this DIC, so you'll have to remember this DIC is that small peptide which I was talking about. It should not remove dynein or dynactin from the phagosome. Now, when I say LPs, remember the late phagosomes which were moving towards the center of the cell. And why we chose this is because we want to study dynein-driven motion. So therefore, we choose LPs because they move towards the center of the cell. Okay, so what we first figured out, and I'm not showing you the data for this, is that if I add this peptide, it does not remove dynein or dynactin from this phagosome. Because if it just simply removes dynein or dynactin, there's nothing left to study, right? So that does not happen. I'm not showing you the data. The second thing, is that we had shown earlier is that the dynein is localized very deep. So what this circle that you see is the surface seen in cross section now of a single phagosome. So this diameter is 2 microns here, and you see the surface of a single phagosome seen in cross section, and the green is dynein. Imagine it to be dynein, and you are fairly uniform on the surface. But if you look at the late phagosomes here, the distribution of dynein is not uniform, it is punctate. And what we had shown is that the dynein clusters into small lipid raft-like domains on the surface. And this is what biases this cargo, makes this an unfair coin, and makes you go towards the minus end of the microtubule. So when we added DIC, which is supposed to go and stick to dynactin, means to regions where Dynein-Dynactin cargos are present. What you actually see is fairly nice, is that on the early phagosomes, this DIC goes and sits everywhere, but on the late phagosomes, the DIC sits in these kind of punctate regions, and that is great because you know that the dynein is in this puncta, so DIC is going to the right place. Okay, so then we ask that what is the function of this attachment? Okay, now what we did is that we made this DIC, this little peptide here, which was either a normal form of DIC or a mutated form which cannot bind dynactin. Okay, so that is a famous negative control in our assays, in biology rather. And what we did is that, you remember these microtubules, okay? So we purified a lot of microtubules and we simply took an extract from brain, okay? What does brain extract have? It has dynein and dynactin, and we added these brain extract to microtubules, let it be for a while, and then you simply do centrifugation. And when you do that, all these microtubules clump together and pellet down, and whatever dynein or dynactin is bound to the microtubules comes down with the microtubules. So by doing this, you can assay the binding affinity of dynein dynactin to microtubules. Now, here I want to make a distinction, which is, and this distinction is normally not possible to make in other kinds of assays. I will try to show you how we have tried to do this. Your ability to bind to the microtubule, if you are a dynein or dynactin, depends on two factors. One is the on rate and the other is the off rate. Okay, so this experiment where you bind this brain lysate to the microtubule is done in the absence of ATP, and it is known that in the absence of ATP, once you bind, you don't come off. Okay, so this experiment essentially measures your ability to bind, which is K on, the on rate. So when we do this experiment, so this is the real one. Okay, this is the wild type, the one which is supposed to interfere with this binding here. You see that both the amount of dynein and dynactin is reduced compared to the one which is supposed to do nothing. Okay, this is the mutant one. So this, because the K off is anyway very low, this assay essentially measures the on rate, and you see that the on rate is reduced when I disable this interaction. In other words, dynein dynactin is having starting trouble. Okay, K on reduced when I disable this interaction. So this interaction is, so how do we assay this further? So to directly measure this in a more functional assay, what we did is that we did the same experiment here, and you simply hold individual phagosomes in an optical trap and hold them over the microtubule and score whether a motion is detected or not in a given period of time, and we call that the probability of motion. So you can calculate a probability of motion, and what you see is that when you have this, the actual dynein, the peptide which is supposed to interfere with this, the probability of motion is significantly reduced. Okay, which agrees with this finding here that the on rate has gone down. Okay, because he's just not able to move. But the question is, what happens to the off rate? So it's important to distinguish between these two. The on rate is reduced, well, fine, but once it gets started, does it do equally well? To do that, what we simply measured is this duration for which the dynein-dynactin complex can generate force. Remember, when you are away from the center of the trap, you are actively generating force. So I won't go into the details of this, but what we essentially saw is that this duration, the stall force duration, is no different, is the same for the mutant and the wild type condition, which essentially tells you that the dynein-dynactin interaction, disrupting this interaction, has no effect on K off. Okay, no measurable effect on K off. So this interaction between dynein and dynactin is required for dynein to get started, but once it gets started, it has no problem even if this interaction is disrupted. Next, we move to how much more time do I have? Can the organizers tell me?
Yes, 15 minutes.
Sorry?
15 minutes.
Okay, great. I'm right on time.
There is
Okay.
another hand raised, so can you take that question?
Yes, yes, absolutely.
Yeah, please go ahead. Markand? Where is he?
So somehow I can't see these hand raises.
Sir, I accidentally touched it. I don't have a question.
Oh,
Sorry.
okay,
Okay.
no problem. Yeah, okay. Okay, so yeah, right. So just to kind of summarize this, when you disrupt this interaction, the whole machine has no problem in getting has a problem in getting started. But once it gets started, it works fine. So now let's look at the other interaction. Okay, so this is the one which we already studied. Now we come to this one where why does dynactin have to bind to the microtubule separately when dynein is already binding here? Okay, so this interaction you disrupt by adding an antibody. Again, this this strategy has been adopted earlier. I showed you in the previous slide by other workers, but here the difference is that we are doing it on a real cargo and are able to assess the what happens as a result using an optical trap. Now again, we tested that this treatment does not remove dynein or dynactin from the phagosomes, which is important. So now when we do this experiment, again, what is this experiment? You take microtubules and you add dynein or dynactin to it, and you pellet the microtubule just to see how much dynein or dynactin comes down. If more comes down, then there's a higher binding affinity. Now, this binding affinity is a composite of the on rate and the off rate. Okay. So here what we see is that when you do this experiment in the absence of ATP, where you know that the K-off is low and all you are measuring is the on rate, whether you add the real antibody which disrupts this or you do a control antibody which is supposed to do nothing, there is no difference in the amount of dynein or dynactin that comes down along with the microtubules. Now, this is very different from what you saw earlier where we had disrupted this interaction and you were seeing that with the DIC, the amount of dynein or dynactin was significantly reduced. Okay, so this means that dynactin has no starting trouble. Okay, if you disable this interaction, it can attach to the microtubules fine. And this is actually what we also measure here, where how to get rid of this. Yeah, what you see here is basically where the probability of in a given time interval, what is the probability that something will happen if you hold this phagosome above the trap is, there's hardly any change. So there's no problem with the on rate. Now let's go to the off rate. Here again we are measuring this. So you remember what I had told you here. We are measuring this duration. Once you engage, how long do you stay on? The inverse of this time is simply the off rate, right, of a net team of motors. And what you do is that you measure this, hold on. So can you see the entire screen or do you see this? Why this? Okay, right. So when we do this experiment, when we measure the time for which it can stay on, we see that when you add, shown in red here, when you add this antibody, it is significantly reduced. This so the take home from this is that here are two parts of dynactin, of the Dynein-Dynactin machine. One is this where dynein and dynactin attach. One is this one where the microtubule and the this dynactin attach, and they have very different functions. This guy allows the Dynein-Dynactin machine to engage to microtubules quickly, and this actually agrees with this Cryo-EM data which I was showing you earlier, is that what people believe is that the dynactin orients dynein, possibly by virtue of this interaction, the right orientation so that it can catch the microtubule quickly and start walking along it. But this interaction has, once you attach, doesn't seem to do much. Okay, then you can move fine even if you're not attached here. So in contrast, this interaction here where the dynactin is binding to the microtubule, is not required really to get started, okay, but it is required to sustain the force when you are moving. Now, how might this happen? Okay, so this is a speculative model, and this is something from a physicist point of view, which is interesting to understand. Try to understand. Here is the whole machine. Okay, it's a cartoon, of course. Here is Dynein in blue, and it wants to go this way. Okay, and it is attached to a cargo via Dynactin, which is this, and Dynactin is again attached to the microtubule here. Okay, now when Dynein pulls in this direction, and let's say I pull with the optical trap in the opposite direction, you can expect a torque to be generated, and this is shown by this arrow here. When this torque is generated, this part would push down, and if this object, this is the part of Dynactin which binds to the microtubule, is bound strongly and it refuses to slip, then this torque can be this push because of the torque can be sustained. If this slips, then you cannot sustain force because this angle theta, if imagine this slips, imagine trying to walk with a stick. Okay, this slips here, this angle theta shown here between the Dynein head and the microtubule will become very small, okay, because you will just tilt in this direction. And when that happens, there are actually strong electrostatic bonds, which are orientation dependent electrostatic bonds at this place, which I did not talk about, which might get disrupted. So to maintain a correct orientation is important for Dynein, and this has been widely accepted. So we believe that there is this machine where this interaction between the Dynein and the Dynactin via this Dynein intermediate chain is important to get it started, and this is once you get started to stay on. Now, I think it is important to appreciate here that we believe we are able to dissect what functionally this kind of a nano machine does, what different parts of it do to sustain force generation inside cells. And this is important for many kinds of force generating events if this turns out to be true. I think I would like to keep about 5 to 10 minutes for discussions if people have questions. So what I did not discuss today is that we can now assemble these kind of Dynein-Dynactin and actually kinesin complexes on supported lipid bilayers. So, I mean, if anybody is interested in working on this kind of experiments, you are most welcome to contact me, where you can assemble cargoes of all kinds of complexity in our lab now. And what we now wonder is that if you are a phagosome or a cargo coming down this road and you see an obstacle like this here, what will happen? So we can engineer these kind of things also. We're in the process of doing that. And now you have to think of this optical trap as an obstacle here, right? But there are real obstacles inside cell, let's say other cargoes or other proteins stuck to the microtubule. What is the effect of that? And that is really relevant to many kinds of neurodegenerative diseases, something that we would like to understand in my lab now at IIT. So I will stop there, and thank you to the organizers for allowing me to speak on this forum. And if there are any questions, I'm happy to take the same.
Yeah. So questions from audience? So there are 2 questions in chat.
Okay.
So Vandana, can you read your question and ask Professor Mallik?
Hello, sir. Sir, actually, I want to know that you have shown me a movie that there is a one complex of Dynactin and Dynein motors which carry this cargo, this common LP, the late Phagosomes.
Yeah.
I want to know that how this load has been shared between these two different motors. Are they equally shared? And I also want to know that what is the role of Dynactin in this whole process. The most of the things you have cleared later in the later slides.
So about the sharing of the load, there is a very detailed work. I think Ambarish will talk about that later today. About load sharing. Most of this is computational work. It is difficult to experimentally measure how much of the load is with shared across the motor. So A lot of this is computational work. I think Ambarish is better qualified to talk on that. So please listen to his talk later in the day.
Okay.
I think his talk is today later. So what was your second question?
Second question, you have actually given the answer in between, but the last one actually I want to know, like, how, if there is a no dynein, how the individually these dynactin will behave.
If there is no dynein, how dynactin will behave?
Yeah, I mean it's like separately how what is the how it behaves, like
No,
we are not talking about any complex and any late
right.
Ferguson. It's general way, yeah.
Right. So what is known is that people have purified dynactin separately, okay,
Yes.
and they have attached it to beads and done other kinds of assays where more or less the conclusion is, I mean, if you send me an email, I can send you the relevant papers, but the conclusion is that this dynactin more or less skates along this microtubule, and that's why this picture was shown like this, okay. It can't generate force by itself because it can't hydrolyze ATP, but it skates along this microtubule, and people think that sometimes it acts like a brake, sometimes it activates dynactin. So all these kind of functions have been ascribed to dynactin, and people try to interpret in terms of how it may help dyning.
Okay, thank you.
But Roop, a continuation of with this, I have a question.
Yes, please.
If the dynactin is alone, of course it has some contact with the microtubule, but it will simply diffuse, isn't it?
It will. So you don't see, I mean, people think there are independent functions of dynein, but not much is known about it. It will stay on for a while and then it will diffuse. You're right, absolutely.
Okay.
And it will have some on time, and then it'll fall off.
Yeah, but before that it will not have any directed movement, right? Because it doesn't have
any. No. It will have some Brownian motion. Yeah.
So basically, I have one simple question. So how are these proteins are organized on the phagosome? That one, because this organization is also important.
It is important. Yeah, it is important. So I had shown you one slide to try, but I didn't want to go into the details. 1 minute. So perhaps I have this slide, so I will show you. Yeah, here. So from what we see in this picture, imagine if green is dynein. On the early phagosomes, it is distributed more or less uniformly. You can see this, okay? But on the late phagosomes, they are organized into these kind of little clusters. That's what we know at present. And these clusters are important because you can have high local dynein density here, okay? And how exactly all these are controlled is a very exciting different area of research. We think that cholesterol has a big role to play in this.
These are only dynein, not other species are there.
Yeah, this is only dynein that you are seeing here. Correct. I mean, it's very difficult to give a complete answer to how hundreds of proteins are organized at the microscopic scale on such a cargo.
Yeah, so there is another question from Aritri. So Aritri, can you actually ask the question?
Hello, sir.
Yeah.
I just wanted to ask whether every time dynein and like it binds to the microtubule, is it always necessary that Dynein-Dynactin complex also does the same? Like they have two separate binding sites. What influences their binding together with the microtubule? Is it possible that they can have two different attachment rates to the microtubule?
It is very much possible, and I think that is actually what happens. But once you have it in a complex, all you can measure is the net combined rate of binding, right? So either you measure a net combined rate where it's difficult to tell whether dynein bound or dynactin bound, or you measure them separately where you know that one is not affecting the other. So when you have the entire complex, so it's hard to tell whether dynein has bound or dynactin has bound. Presumably both have bound, that's why you are moving. But that's what we try to do in a small way, where we try to disrupt this interaction and see is there a problem, okay? And the problem seems to be that if this interaction is gone, the binding of dynactin to microtubule is gone, then you will attach, but then you have you can't sustain the force. That's what.
Yes. Yes. Okay, sir. Thank you.
Sure.
Any other question?
I think we are running out of time also, so we can move on
No,
to
no, actually yeah. No, we have some time still.
Okay,
We
sure.
have another half an hour for
Yeah.
discussion.
Okay, okay.
For discussion, actually, we have another half an hour. So another question actually I have. So at some point you have discussed that the more than 6 number of proteins are required. So when you are calculating these probabilities,
Yeah, I will go to that slide. 1 minute. Yeah.
why
Yeah.
the number 6 is important here?
It's not important. It's what you measure, actually.
Okay.
That's what it is. Okay. So you measure the force, and so this is the histogram of forces. Okay. And for late phagosomes, which is what we are studying here, you see that the forces are always 4 piconewtons or more. Okay. With red bars. And we know that a single dynein generates about 1 piconewton.
Correct.
That tells you that you have usually about 6 or more dyneins working together if the forces are assumed to be additive.
I see.
Yeah. So that's why we take 6 or more, because that's what you usually measure on this. And here so the dynein force in this case is about 1 piconewton, but the kinesin force is about 5.5 piconewtons, 6 piconewtons. So you have 2 kinesins, and this is a separate area that somehow nature has decided to pit 2 strong kinesins against multiple weak dyneins, and that in itself gives a much more rich phase diagram about how this system might work. And that's people have tried to address that by computational studies, and perhaps you will hear about that from Ambarish also later in the day.
Yeah. Thank you. So any other question from audience?
I have a question. You see, dynein has dynactin to assist in increasing the processivity. Now, evolutionarily, if I understood correctly, these are not A plus ATPase or something like that, right?
Yes.
So among others who are in the same category, is there any evidence that they also have such, assisting, analog of dynactin?
Good question, Debashish. Offhand, I don't know of any other dynactin equivalents which work for other ATPases, but I should look this up a little more carefully. But as of now, I have never heard of a dynein equivalent for another AAA ATPase. That's what you're asking, right?
Yeah.
Sorry, a dynactin equivalent.
Dynactin equivalent.
Yeah, dynactin equivalent. No, I have not heard of that actually until now. No.
So maybe specifically it evolved just to assist
this dynein, perhaps because you need much more processive motion here compared to other ATPases. It is possible. I really don't know.
No, because there are some helicases also who have probably similar structure, right?
And I think the helicases are also quite processive. Is that correct, Debashish?
Right. Without something like dynactin, right?
No. So now when we are talking about helicase versus this, of course the distance between the binding sites for DNA will be very different from on microtubule, correct? So if we talk in number of steps rather than in distance moved, so for a typical dynein, it will take about, let's say, 100 steps before it detaches, a dynein-dynactin complex. What is that number for helicases?
I would guess helicase, it would be much more, much longer.
Much more. Okay.
So that's my guess. Okay.
Okay.
I don't remember the number.
I see. If that is more, then maybe you need something. Yeah. I don't know what it is.
Maybe because it encircles.
Yeah, so it can't diffuse off. Yeah, it's like
that
a
ring,
like a thing. Yeah, it has nowhere else to go once it detaches. It will reattach again. Possible. Yeah.
That is the trick. Yeah. Okay.
Yeah.
Okay. Thank you.
Any other question?
Okay, if there are no other questions, maybe we can stop here then, in that case.
Yeah. So.
Or I mean, I can stay on as you wish.
Yeah.
Okay, so if there is no other question, then we thank Roop for.
Thank you very much. I think it was a good discussion. Nice. Thank you very much. Yeah.